Hello friends, again welcome to my channel. Today we are discussing about one of the important plant virus, which is the TMV virus, that is the tobacco mosaic virus. Now this tobacco mosaic virus, it is responsible to infect the tobacco plant and other plants which are from the family Solanaceae. That is all of the plants which are from the family Solanaceae, this can be get infected by this TMV virus. It is an important plant virus and it is also the virus which is to be firstly discovered in 1892 by the Dmitry Ivanovsky. The scientist Dmitry Ivanovsky firstly discovered this virus, and in 1935 the scientist Stanley firstly isolate these viruses, that is the TMV virus, in the crystalline form. Now whenever we are describing about the characteristics of this TMV viruses, this TMV virus, it contains the single stranded RNA as their genetic material. That means it is an RNA virus with the helical symmetry. It is a structure of the TMV virus within which you can identify here it is a genetic material which is to be present, and it is nothing but the single stranded RNA. It contains about the 6400 nitrogen bases within this single stranded RNA, and majorly it forms the helical symmetry. The capsomeres which are responsible for forming these capsid structures, these are about the 2130 coat proteins. These are responsible to form this helical capsid of these viruses, and these are responsible to encapsulate this single stranded RNA within them. Here it is a rod shaped structure which is to be present, and preferentially the length of this rod shaped capsid or the virus, it is about 300 nanometer. It is about 300 nanometer, or in other words we can say that it is a 3000 Armstrong, one and the same. Now again, these are the here within this, during the formation of this helical symmetry, you can observe that here these are the turnings or the turns of this RNA, these are get formed. So majorly within this one virion there is the presence of 130 turns. These are the 130 turns, these are present, and within each of these turn, it is the one turn. Within this turn there is the presence of these 16 capsomers. 16 capsomers means in this way you can observe. These are the 16 capsomers, means within each of the turn such type of the structure it is to be present. These are the general characteristics of the TMV viruses. Now whenever we are switch over to the infections or the symptoms that are caused by this TMV viruses, majorly these are responsible to form the mosaic like structures. Mosaic like structure on the leaves of the plants, means the main target for this TMV virus, it is the leaves of the plants, and preferentially the young leaves, these are mostly get affected by the attack of these TMV viruses. Now whenever we are considering or concentrate on the symptoms of these TMV viruses, within that majorly there is the formation of chlorotic regions. Chlorotic regions, these are get formed, and again there is the necrotic lesions, these are also get resulted. Now for the entry of these viruses inside the plant cells, it is carried out through the simple type of the wounds which are present on the surfaces of the plants. Through that wounds, these viral particles, these can get entered within the plant cells. Through the appearance of wounds or through that wounds, there is the entry of viruses. Here the whole virion particle, it enters the cell. It enters the cell as it is a single stranded RNA. Preferentially, firstly this virion particle, after entering the plant cells, there is the disassembly of this virion particle and the viral genomes, these are get transported within the nucleus of the cell. and within that there is the replication. There is the replication and the multiple copies of the virion particles. These are get synthesized inside the nucleus. After that, these RNA genomes, these are get transported within the cytoplasm. These are get transported within the cytoplasm, as it is a single stranded RNA which is to be present as a genetic material. So preferentially it can be directly acts as a messenger RNA. Okay, as it can act as a messenger RNA, there is the translation of these RNA molecules within the cytoplasm, and there is the synthesis of the coat proteins. Translation and the synthesis of the coat protein. Synthesis of the coat protein. Now, due to the synthesis of this coat protein, now there is the assembly formation. After that, there is the assembly formation. Assembly formation, and within a single cell, up to the 200 virion particles, these can be synthesized. Up to the 200 virion particles, these can be synthesized. Means it can be considered as a disease cycle of the viruses, within which firstly through the simple wounds there is the entry of these viruses inside the host cell. From that there is the disassembly, that is the uncoating of the virion particle, and after that there is the transportation of the nucleic acid of these viruses inside the nucleus, where there is the replication mode, through which there is the multiple copies of the viral genomes. These are get synthesized within the nucleus of the host cell, that is the plant cell, and after that these are get transported within the cytoplasm of the cell, within which there is the translation process, and through the translation process these coat proteins, these are get synthesized, which is essential for the assembly formation, and these virion particles, these are get new progeny virions or the virion particles, these are synthesized within that cell. Now here about it is the restricted region, that is one cell only get infected. Now it is the primary inoculation of the viruses, that is within that primary inoculation, these primary symptoms, these are get developed, that is majorly there is the formation of the yellow veins on the leaves, and preferentially these primary symptoms, these are only for the shorter time period, these are not constant one. Whenever there is the progression of the disease, these primary symptoms, these are get disappear, there is the disappearance of the primary symptoms, and majorly as these viruses, that is the plant viruses, majorly these are attacking the leaves, majorly there is the destruction of the chlorophyll pigments also, and there is the formation of the yellow veins or the spot like structures, these are get developed onto the surfaces of the leaves, which are developed into the different types of the symptoms, and majorly immediately after that, as it is highly contagious, it is also spreading at a higher rate. Now inside the plant cell, majorly for the purpose of transportation of these viruses from one cell to the another cell, within that there is the role of an important protein of this TMV virus, which is to be called as the movement protein. Movement protein, and majorly these are transported from one cell to the another cell through the plasmodesmata. That is, these are the connecting regions in between the two cells, through which there is the transportation of these virions with the help of their movement protein. These are synthesized their movement protein, and this movement protein, it is essential for their transportation or spreading from one cell to the another cell. Means within a single cell, whenever the maturation of these particles, these are appeared, after that through this plasmodesmata with the help of this movement protein, these are transferred to the nearest healthy cells to infect that cells, and by this way there is the development of the from the localized region. Now there is the disease, it is get progresses towards the systemic infection. Systemic infection, that is the different regions of the plant cells, these are get affected by this TMV viruses. So here it is the information about symptoms of these viruses. Now, how there is the majorly some type of the characteristics of this TMV viruses. It includes majorly these viruses, they can be remain alive within the dormant state. Within the dormant state. Dormant state means they are not growing but they are alive. These are not get killed. Means within the plant debris they are present within the dormant state, but whenever they are in contact with the live cell tissues, live cells or the tissues, at that time they are again start to grow. So majorly these are not easily get destroyed. These are not easily get destroyed. These are also withstand the desiccation conditions. These are get inactivated at 90-degree temperature. Means also these are also the temperature resistant also. So these are the specific characteristics of this TMV viruses. Along with that, majorly these viruses, these can remain active or these are remain alive whenever we are storing these at the within the laboratory at 4-degree temperature up to the 50 years. Means these are highly stable in nature. These virion particles, these are highly stable in nature. Okay. So it is about the symptoms, the characteristics of the viruses. Now, how these viruses, these can be get transmitted. Now, the major source of the transmission of these viruses, it is majorly the sap of the infected plant leaves. Whenever there is the sap of this infected plant leaves, it is in contact with the healthy plant, then through that there is the easy transmission of these viruses to the healthy plant. Then the different mechanical workers who are working within that regions, and if they are using the cigarettes or the tobaccos which are containing these viruses, through that also again there is the transmission of these viruses. Then the seed coats of these tobacco plants, these can also be get contaminated by these viruses, and once these seed coats these are get contaminated, then the plants which are originate from that seeds, these are also get infected due to that contaminated seed coats. Again, through the mechanical, there is also the mechanical transmission of these viruses. The different equipments that we are utilized with during the farming, at that time, the same equipments, if these are get utilized for the healthy plant and also for the infected plant, then through that again there is the transmission of these viruses within the healthy plant. So these are the different transmission modes. Okay. Now the last and important thing about these, it is the control measure. How to control the spreading of this TMV virus, because as it is responsible to infect this tobacco plant and the different members of the Solanaceae family, most of the plants from this family, these are of economic importance, and if once they are get infected by this TMV viruses, as it is highly contagious, most of the yields it is get destroyed through that infection. So it is very important to develop the different control measures for the controlling the spreading of this TMV virus. So within that, preferentially whenever there is the preparation of the seed beds, these are from the large distance from that of the warehouses where there is the presence of this debris of plant debris. Then there is also complete destruction of the infected plant debris by the burning, because the burning, it is the only procedure for the destruction of the viruses, because if the plant debris of the infected plant, it is remain as it is, through that within that these plants, these viruses, these are remain alive. Okay. Now along with that, majorly it is tried to develop a resistant cultivars of this tobacco plant, to develop the resistant varieties of this plant which can resist the growth of these viruses within them. These are the some control measures to control this TMV virus. So here it is, within short, it is an information about the TMV virus. So if you like this video, please subscribe my channel and also share this video with your friends. Thank you.